This is the inoculum preparation procedure for MLRS STaTAST. First, a few single-type colonies are picked from the agar plate using a sterile loop. These colonies are then inoculated into the peptone water. Patient samples, like urine, can be directly used as the inoculum, provided there is microscopic confirmation of the presence of more than 5 bacteria, uniformly distributed per oil immersion field at 100x magnification. The optical density of the bacterial suspension in peptone water is then measured and recorded in McFarland values. The value must be greater than 0.5, as lower inoculum suspension might lead to longer incubation time. MLRS STaTAST is based on CLSI broth microdilution method. The antibiotic, according to CLSI breakpoints, are coated onto microtiter plate with specific panels for gram-negative and gram-positive bacteria. Using a single-channel pipette, 200 microliter of sterile fluorescent culture medium is added to the negative control wells, A1 and A2. Using a single-channel pipette, the bacterial suspension is mixed thoroughly. Then 400 microliters of inoculated peptone water is added to the fluorescent culture medium. The inoculated culture medium is mixed gently. The medium is then transferred into the reservoir. We use a 12-channel pipette to transfer 200 microliters of the inoculated culture medium into each well of the antibiotic-coated plate. All the wells in rows B to H are filled with 200 microliters of the medium. This process takes only 30 seconds for each sample, which is a huge time saver compared to the traditional disc diffusion method, where the laborious culture streaking can take about 5 minutes per sample. The tips from the first 2 channels are discarded and the wells from 3 to 12 is filled in row A. A3 well is the positive control well, a well with no antibiotic. The plate is then incubated at 37 degrees Celsius incubator.
This is the semi-automated antibiogram device. The semi-automated antibiogram device is connected to the computing device which runs the imager software. The tray on which the incubated antibiotic plate is placed works with the touch of a button. Once the incubated antibiotic plate is fed into the antibiogram device, the type of bacteria is selected if known. If unknown, the generic type is selected. The sample ID is fed into the device and the image is processed by pressing the Acquire Image and Analyze button. The image is captured, processed, and the result is generated and displayed with 2 distinct color boxes. The resistant box is displayed as a red box and the sensitive box is displayed as the green box. The result also is displayed in the form of an Excel sheet in which the MIC is determined. The database, which can be linked with the laboratory information system, is also integrated into the software.